Hello everyone, my name is Lauren Chao, and this summer I did research with Dr. Gretchen Meyer and the Meyer Integrated Muscle Physiology Lab, and what we wanted to investigate was whether or not fat can affect muscle regeneration. So typically, the biggest issue people face after injury or disease is the recovery time it takes for their muscles to heal. And when we think of muscle regeneration, typically we commonly see fatty infiltration, and especially in individuals who are obese, as the excess fat as having negative effects on muscle rehab. So the way that muscle grows is that myoblasts go through myogenesis and they mature and grow again. So we wanted to know if we could ask this question in a dish, and our hypothesis was that fat will inhibit myogenesis. So the way we did this was essentially, the way we did this was we grew adipocytes, we collected the media that they secreted, and we placed them on myoblasts to see whether or not they would grow. And here is our experimental setup. So we had two separate timelines, and we first plated adipose progenitor cells, and we placed them in different media, such as adipogenic induction media, to get them to differentiate into adipocytes. And the way we could see that they differentiated is that they formed little tiny lipid droplets within the cell. So the same time we were growing the adipocytes, we also wanted to grow our myoblasts, so they'd be confluent enough by the time we placed the DMEM media onto our myoblasts. And after we did that, we wanted to see and quantify whether or not our myoblasts differentiated into myotubes, and our DMEM was placed on our myoblasts, and then we wanted to see whether or not our myoblasts differentiated, and you can see in the green, these myotubes represent that, or if they did not grow. So after we took the media from our adipocytes, we ran 4 analysis assays, and our first was staining our adipocytes and quantifying them with oil red O, strictly just to see that they did in fact differentiate. And then the next assay we ran was a quantikine assay, qPCR, and then staining and imaging our myoblasts to see whether or not they did grow or not. So this brings us to our first step of analysis, and here you can see that in our growth media we had no differentiation in our adipocytes compared to our adipogenic media. And these red adipocytes show the lipid formation within the cell. And we quantified this in a graph with the percent oil red O area in each of these images, and we did see a high rate of differentiation in our adipo-treated cells. And one of the things that we also asked was whether or not a dose response, so the amount of differentiation of each of the fat cells, so if we had a plate with low differentiation versus a plate with high differentiation, if that had any effect on muscle regeneration as well. So this brings us to our next assay that we ran, and we wanted to see the components inside each of the secretions of our adipogenic treated cells. So the way that an adipokine assay works is that we had 38 specific adipokines commonly found in fat cells, and we placed the media on 4 different templates. We had our growth media and then our adipogenic treated cells. We placed the media on, and then the intensity of the dots showed us whether or not that specific adipokine was present. And we noticed 3 key differences between the adipogenic maintenance media and then the growth media. And the first one was the presence of resistance. So I don't know if you guys can, this one was very stark, so the intensity of these dots was much higher than the growth media. Then we also found that IGF-BP5 was present in the adipogenic compared to the growth media. And then lastly, PREF1. This one wasn't as intense, but we still wanted to confirm through qPCR later whether or not these proteins were in fact in our original set of DNA. So here we have a quantification of the fold change of each of these proteins in our adipokine assay, and we see that resistin was a very high fold change, close to 60, and we also see that IGF-BP5 and PREF1 was also increased. So this brings us to our qPCR, And like I mentioned before, we wanted to test the dose response as well. And we found that wells that were treated with high rates of differentiation and low rates of differentiation, that there wasn't much of a difference. So as you can see, after we ran it, our resistin didn't really show up, which was sad, and our IGFBP5 showed that low actually had a higher rate than the high differentiation. Pref1 had a higher rate of differentiation for the high adipocyte differentiation rates, and our PI also was interested in follistatin, so we ran a test on that, and we saw that there wasn't a difference between the different dose responses. So I told you guys about our adipogenic side of our experiment, so this brings us now to our myogenic side. And this is our plate layout for our myoblasts. So in each of these cells, in each of these wells, we had myoblasts that we plated and grew, and then we placed different medias on each of the wells to see whether or not they would grow. And sadly, our anti-follistatin columns did not. They died. And we just tested our low differentiation rates, high differentiation rates, and our growth media compared to our blank. And we saw, after a couple days in the media, that our AMM group did in fact grow a lot of myotubes. And as you can see in each of these pictures, the green represents our myotubes, which show that the muscle myoblasts matured. It stands for the myosin heavy chain, and then the blue is the nuclei of each of the cells. And as you can see, we ran a quantification analysis, and we basically calculated the percent of area that the myoblasts or myotubes took compared to black space. And we saw that, again, there wasn't a difference between the lower differentiation rates and the high differentiation rates of fat. But we did see that the adipogenic treated cells had a much higher growth rate than the growth media and the blank. So this ended up disproving our hypothesis that we originally thought that fat promoted myogenesis, but it in fact promotes myogenesis. So we hopefully are going to investigate this further and possibly make more therapies to help with injury patients in their muscle rehabilitation process. And I want to thank you guys all for listening, especially my lab, so kindly came, and Jim and the uSTAR program. You guys have been fantastic. I had a great time. Any questions? Yes?
Yeah, do you guys, I thought this was really interesting. Do you guys have any ideas as to the mechanism behind why this adipose tissue increases myogenesis?
So that's just specifically what we were trying to figure out. So we tried to test whether or not the certain proteins inside the adipogenic media and the adipose tissue actually increased the muscle growth rate. And then the part of our setup that we also wanted to test was if the cells surrounding our differentiated cells also had an effect in helping muscles grow, but we found that it was the adipose tissue cells. Any other questions? Yes, Jim?
So there are three different types of fat: white, beige, brown.
Yes.
And so you have that fat promotes muscle growth or regeneration at the conclusion. So that's cool. Do you think it's kind of cell type specific, or like all three types of fat?
So our original layout was testing brown fat, but it was unsuccessful, so we switched it to just fat in general. But my PI has found that brown fat does promote muscle regeneration, especially follistatin she looked at, and we found that it did increase, but my results were giving me a hard time, so I did not investigate that further.
But with your results showing that fat promotes muscle growth, was the fat what type of fat were in your experiments? Was it white? Was it beige?
Beige fat.
Beige. Okay.
Yeah. Any other questions? Thank you guys so much.